All right, welcome everybody to the second of our webinar series, Indicator versus Pathogen. Just want to thank everybody for taking the time. And if you guys have any questions today, please do ask. There is going to be a Q&A section on the bottom or on your Zoom page, as well as a chat feature on your Zoom page. So go ahead, and whenever you have a question, we will try to get those answered in real time, or wait until the end if that is appropriate. So again, we do suggest you ask those questions. We look forward to kind of seeing what you guys are seeing in your plants and in your labs, and anything specific to your needs, please do ask. Also letting you guys know that this session will be taped for others to view. So if you have somebody that couldn't make it, or another section of your plant that wants to view it, please contact us, and it is available to be viewed at a later date. So from the Cherney team, welcome to the webinar. So Matthew Fensky, our business development manager, is going to be doing a majority of the speaking today. So if you have questions for him, please let us know. My name is Robert Brown, the client services manager here. We are lucky enough, we have two of our technical team here as well, Michelle and Allison from our lab, and they will be happy to answer any questions that you guys do have. So please throw any questions that you have, and we will get them answered. So thanks again, and I'm going to throw it over to Matthew Fensky.
Thank you, Robert. So let's get started right away on what the heck are we looking at today with indicators versus pathogen. What are the tests that we're doing with those? What do they tell us? And what are we using those results for? Or is it just another checkbox to have? I'll tell you the answer to that one right away is no. They definitely give us a lot of opportunities to look at our product and our environment and really give a good, safe product with high quality to our customers. So when we break this down, we really have our two items, the indicator organisms and our pathogens. Our indicator organisms is really looking at count. How many bacteria are there? Not necessarily bad bacteria, but the bacteria that are present that we are targeting. As we look at that, we can use that as more of a leading indicator within our environment and our product to say maybe it's not out of spec or there's no pathogens present where there's a risk, but ultimately we still can say that something's going on. We're seeing an increased count on our indicators, such as an APC or Enterobacteriaceae count, that's going to allow us to take some sort of action to get that back into check before it does go to present a problem. So more of a leading indicator, especially when we're talking product and the environment. A little bit on the back side of that, especially on product, is the pathogens. So when we have pathogen testing in product, obviously our product's already made. If the pathogen's present, now we have risk if it's out into the consumers for a recall or to worse hurt or harm one of our consumers. So a little bit on the lagging side, similar in the environment, we may see that pathogen present. The nice thing with the environmental monitoring that we talked about last time was that it still gives us a proactive approach to be able to look at where we've seen pathogens in the past and take care of them in the environment before they ever get to our product. So to answer that original question, both of these are going to be a very key role in our testing programs, both in the environment and our product. So we're going to start by talking about indicator organisms. For those of you that joined us on the environmental monitoring webinar, this slide may look very familiar because it does play such a huge role when we talk about our indicators. So an indicator organism is a bacteria that's going to grow in the same set of circumstances with the same growth environment as those bacteria that could be harmful to us, such as Listeria, the E. coli. The indicator organism itself, not inherently dangerous, but it might be a quality check and definitely give us that opportunity, like I'd said on the original slide, to look at what our microbial, or using the food safety system terms, what our biological risks are for the site, for our product, and our overall organization. So you can see there some of our most common indicator organisms, when we start looking at E. coli, Salmonella, Listeria, is the coliform groups, the Enterobacteriaceae, to kind of spread that out a little bit further, and we'll talk about this, and look at where the Salmonella lies in there. Listeria, so Listeria monocytogenes, we talked about last webinar, is kind of the wimpy kid of the Listeria family, or genus, excuse me, and will be the last one to grow, but it's going to grow in the same environment as what the rest of the Listeria grow in. So by testing for the whole species of Listeria, we can see that our growth environment would be correct for monocytogenes. So some of our most common indicators, and you may see these in your facility, be doing them yourselves as testing or sending them out to us or for testing as well. But we talk about aerobic plate count as, especially from like a clean equipment swabbing, and that overall microbial load. So most of our bacteria that are present are going to grow in the presence of oxygen, and that's what this aerobic plate count is looking at. Thing to keep in mind here again is on an APC, that is good bacteria and bad bacteria, so that overall bioburden. When we start talking in products, sometimes we have the lack of oxygen, and that's where we're going to go ahead and do the anaerobic plate count. I'd mentioned earlier about looking at E. coli and the risks that are associated there. And we'll talk in depth on the coliform here as our example or case study, but those coliform counts to really understand how safe is our product, what is our quality of our product, do we have gassing issues that may come out down the line, and so on and so forth. Taking a broader approach of that same area is that Enterobacteriaceae. So we'll see in some of the visual depictions later that the coliform group that we're looking at as an indicator organism fits inside of that Enterobacteriaceae, or EB for short, and gives us a much broader scope. Then when we start talking about more of our quality issues, what's our air quality that we'll see a lot of times, is that yeast and mold count. Similar things within product. Now, obviously, if we have mold or yeast that we're using to make our product, that plays a little bit different role as well. So when we talk about our indicator organisms, we may be talking more on a quality standpoint with that yeast and mold count and lactic acid bacteria. All the way through, we want to be monitoring these counts and trending it across our platforms in our time. So I said we were going to use coliforms as our case study. What the heck's a coliform? Well, if you're working in the dairy industry or many of the other food industries at this point, you're probably well versed in what a coliform is, but just to kind of go through again, we're looking at a bacteria that's going to ferment lactose. So we may use that in the future as like a color change or indication. But the real telltale sign of a coliform is that within 48 hours, it's going to have a production of acid and gas, given that scenario. If you remember correctly, on the last webinar, we talked about replication. How fast are these bacteria going to duplicate? And we saw an example where every 20 minutes, our bacteria is going to duplicate itself. And that was actually a coliform example. So given the right scenario, coliform are going to replicate themselves every 20 minutes, which really allows us to use them as an indicator very well because they have that rapid growth and we'd be able to detect them at an earlier state. Throughout the presentation today, you're going to hear me say earlier state, rapid platforms, quicker turnaround. I still want to emphasize that when we talk about those things, that's not a 15-minute rapid platform. There's still an incubation period that goes along with that. So typically we're looking out for like the coliforms and many of our other platforms at 48 hours as the quickest turnaround time on those. So with the coliforms, I said that fits within that Enterobacteriaceae family, and it really covers 5 genera. The pathogen that we most notably talk about with coliforms is the E. coli 0157 group that is located within the coliform family. So when we use that as an indicator, really talking product safety, really talking the quality of our product. But there is 4 other ones that fit into the coliform group as well. Again, relatively narrow scope, and that'll be pretty easy to see when we bring up what the Enterobacteriaceae family looks like. So our bacteria for coliforms is going to grow well on various different medias, pretty wide range of temperatures. And that's what allows us to be able to look at this across many different food groups, many different environments, because of that broad range, and really use it successfully as an indicator organism. That's really what it all comes down to, is we want to know, is our product of high quality? Is it safe? Did our sanitation do a good job? And coliforms are commonly used in that standpoint. When we look at where they're from, we talk about feces of warm-blooded animals. We talk about the environment, such as soil, and the water that we're using. Really, when you think about that, one, a lot of that plays into our products, but two, I guarantee you that everybody's environment has some sort of warm-blooded creatures running around in them. I know we got 4 of them sitting in the room here today with us, humans, obviously. So from our standpoint, the other play that the coliform has is our water quality. And so you'll hear a potable water test or safe water test. And that's really looking at the coliform presence or absence within our potable water sources. That'll go anywhere from an ingredient all the way through to using water from a cleaning standpoint. Nice thing about coliforms at the lab, whether that's your lab or in-house or Cherney here, is that they're relatively easy to culture, so we get that quicker recovery and easier representation to be able to see and count those. So looking at our coliform and E. coli, I pulled up our lovely Petrifilm here to really be able to identify, we see red colonies, we see blue colonies that are gassing. That blue colonies, obviously, are E. coli. All of them fit into that coliform group. And then what you can see as the visual depiction here represents is it looks like there's tears or some kind of absence around that actual colony on the Petrifilm. That's that gas production that we've been talking about that really identifies this is a coliform. So we have to see that gas production within the 48 hours to call it a coliform. Now, I mentioned coliform's good. This is what we've used as an assessment historically in the US for years and years and years, really within the dairy industry as well. Coliform has been kind of the staple indicator organism. What's happening is when we look at Enterobacteriaceae, that includes Salmonella, it includes a much broader scope. So the European Union has actually fully switched over to using EB in lieu of coliforms. And we're seeing that trend continue on within the US as well. So if you're not doing EB at this point, don't panic. You're not out of the loop, but don't be surprised as we continue down this food safety journey and continuous improvement that will be the new standard going forward or in the future. Again, we'll take a look here. Because we have our coliforms, which you can see visually depicted here as such a small group within that Enterobacteriaceae genre, we can now look at that bigger picture. Yes, it includes Salmonella, so maybe a better indication of our quality and our environment being clean and acceptable, but it really drives to the opportunity to have continuous improvement. Looking at historic data, and I can speak for the Cherney data, we saw where we were within spec, we were good to go on coliforms repeatedly, time after time after time. And so for us, we still wanted that continuous improvement, and we see this in the facilities that we're in a lot of times too. So we switched over to Enterobacteriaceae testing, looking at that bigger group. Lo and behold, we saw out of spec counts, and it allowed us to do that further investigation and now take our facility one step further in that cleanliness and ensuring that we have a relatively microbiologically clean environment. If we were a food production, obviously, then an even safer place to produce our food. And then just a visual here on violet red bile agar, or VRBA, is our Enterobacteriaceae. So how do we get to the point where we can look at a pretty plate like this and say, okay, here's our count? One of the first things that we start talking about when we talk about indicator organisms is our dilution factors. And really at the root of it, whether it's microbiologically speaking, grape juice or whatever cranberry juice I think that is on the screen as our picture, it's taking something from a very high concentration and diluting it out. And the benefit there from a microbiological standpoint is now it gives us the opportunity to actually see the individual colonies and count them within the countable range. When I talk about countable ranges, those of you that are using Petrifilm, each Petrifilm in the interpretation guide will tell you there's a countable range. So I believe APC is 25 colonies to 250 colonies. And what that's looking at is where are we most statistically accurate that we're going to repeat that if we run that test over again, and that we're actually able to count the colonies that are there. So we're going to look at a picture here in a second where it's overgrown on the plate. There's really no chance to be able to count that, and you're going to see TNTC. We want to do those further dilutions to understand what is our TNTC. Here at Cherney, we want to be able to give you an answer. If your original sample, you say it's TNTC, and we get an actual count, and it's 2 million colony forming units based on the dilutions that we do, you take your remediation efforts, and it comes back, and it's still TNTC. Did you improve? Well, that's where being able to have the dilutions in place really plays a factor. If we go from 2 million colonies as an estimate, down to 100,000 colonies as the estimate, we may still be talking that we are TNTC if we don't do our dilutions. What we just said there, though, is we went from 2 million to 100,000. The remediation efforts that we took obviously are taking in effect and doing what we wanted to do. We just need to take it a little further. We wouldn't know that if we didn't do the dilutions. The concern that comes into play, especially for Cherney as a contract lab, we don't necessarily know what your original count is going to be. And so we don't know what dilution to take or get to take our sample. And the best solution to that is that we'll run it across multiple dilutions. So standard dilutions of 10 and 100, we can go out 1,000, 10,000, 100,000, all the way out. I think we've gone up to 10 million, I believe, if I remember correctly, to actually be able to get that count for one of our customers. So like I said, dilution is going to play a huge role when we talk about our indicator organisms and successfully plating those. So I promised that visual. This is the same sample across a set of serial dilutions on for Salmonella and Chrome Agar. I don't know about you guys, but the first 3 plates starting on the left-hand side, I don't know if I have enough fingers and toes to count that high. Well, I promise you I don't have enough fingers and toes to count that high. So being able to have those dilutions and getting down to that 13 all the way to the right, and then being able to turn around and apply that dilution factor to give us an accurate estimation of what that overgrowth on the left actually is. So very, very key point, and I love that picture depiction of it. So how do we get there? Well, we need to take and figure out what our dilution is actually going to be. For sake of easy math and one of the common dilution factors, we'll talk about a 10 dilution. So when we talk about a 10 dilution, that's taking one part of our sample or original inoculum and putting it into 9 parts of the diluent to get to that full number of 10. Quick cheat on that is to figure out how much diluent you need is that you can always take how much your original sample is and then multiply that number by 9 to get to that diluent needed. So if my original number is 1, I have 9, multiply that by 9, my diluent is 9. If it's 11, I multiply that by 9, 99 of mils of the diluent. So that gets us one dilution. One dilution, as you can see on the plates here, on that previous picture, oftentimes is not enough. So rather than trying to go into a larger number of, or volume of diluent, we can stay with our 9 mils of diluent and go from the original dilution tube to another 9, or take 1 mil out and put it into that second dilution tube that has 9 mils of diluent in it to get an increase of a factor of 10 every single time we're doing that. So you can see 10, 100, 1,000, all the way across the board. Some very key things to keep in mind here when we talk about doing a serial dilution is that we have to have a homogeneous mixture that themselves are evenly distributed in each of those diluent tubes before we go to the next diluent or to the next dilution. So we're going to talk about some things in the sample prep here in just a second, but really making sure that we are vortexing or shaking or mixing those to create that homogenized mixture where the cells are evenly distributed as we go through our dilutions. So as I was saying, that even distribution, a lot of different things that can happen when we do serial dilutions and we start talking about pipetting, the volumes, the blanks, well, for our lab, it's if I'm allowed in the lab and distract somebody, hence I'm not allowed in the lab very often anymore. But we're really going to talk about a lot of this in that GLP webinar. Maybe some of you have signed up for that. I'll put that up on the screen later on, but I encourage you to come back for that GLP webinar to understand this further if you have questions on it. So talked about the dilutions. Sample prep is really going to play a big role in what results we get as well. Our sample prep looks at 3 big things to make sure that we're staying away from them. The sticky cells. So we want one cell to create one colony forming unit. And we'll look at a picture here on the next slide that shows this. We could have 4 cells that stick together because of insufficient mixing, shaking, vortexing, so on and so forth in our sample prep. And that's going to also create one colony forming unit if they don't separate. Now, obviously, we have 4 times the amount of bacteria there than our one individual cell. We want to give you that accurate number. You want to get that accurate number. And that's why mixing sufficiently really plays a key role. We talk about distribution. So if we let a inoculant sit on a spread plate too long and we don't get it spread out, if we use the wrong spreader on the Petrifilm, I know when I was taught about our pour plates, they taught me about swirling, the proper swirling technique. And I was a little bit of an aggressive swirler the first couple of times, and my media actually ended up on the lid of the Petri dish. Obviously, we're taking nutrients away, we're taking that bacteria out of there, and that's all going to start playing a key role into what our distribution is. And then finally, our E. coli and other bacteria will actually move. They're motile. So those swarming cells, we want to be able to make sure that they stay in place. And that's, if we have too much liquid, that's going to spread them because of the liquid. If we have the bacteria that are moving around. So utilizing pour plates, utilizing overlays and Petrifilms to be able to keep our bacteria in place. So I mentioned and promised that picture. You can see on the bottom there, our ultimate goal is to give you that report, that result in colony forming units. How many colonies are there? And that should represent one cell of bacteria. They're single cells, so one bacteria equaling one colony forming unit. And this is where that visual depicts these sticky cells where one cell could equal one colony forming unit. 4 cells could also equal that one colony forming unit if we don't properly mix, and that's what we want to avoid. So we have multiple different methods here. We'll go through here quickly. Pour plates, spread plates, membrane filtration, Petrifilm, and the one that's up here that I'm just going to hit on quickly is our most probable number, or MPNs. So again, this is a quantitative method. We're going to take your sample. We're going to look for that indicator, give you an actual number at the end of it. So we're taking plating or whatever method we're using, incubating part of that, and then giving that resulting number per what the original or what the sample being incubated was. So our example there is 150 CFU per gram. A couple of things to watch for on your C of A's is an asterisk that might be on that. So if we have to go outside of the countable range, that asterisk is telling us that we are outside of that countable range to get to a number. And so that denotes that that's an estimated number. We talked earlier about dilutions. So you may see a less than 10 or where we didn't see growth on the plate. But because there's that dilution factor in there, we can't say 0. We have to go to what that dilution that we looked at was. So oftentimes we'll see that less than 10, less than 100. It just means that we were looking at the plate that had that dilution associated to it. So pour plates and spread plates, they both have their place, they both have their uses. Big thing on the pour plates is that it gives us that extra nutrients. It holds our bacteria solid in one place because we're overlaying our our sample with that agar and letting it harden in the incubation period. On the spread plate, we can put it on top. We can't use as much of a sample, so our sensitivity is a little bit lower there, and then spread it evenly across the top of the dried agar. Excuse me. Nice thing about the spread plate is if we go into some identification or further testing where we want to pull the sample or that colony or that bacteria off, the spread plate's much easier to do so on that. So quick list here, I talked about that being able to plate larger sample sizes with the pour plates, which plays with our sensitivity as well. The nutrients are there to neutralize off on the samples. It's going to hold those motile colonies in place, or those bacteria in place. The one caveat there is that most of that bacteria or the media that's used for that has a real short shelf life, so it almost has to be made that same day. Something to think about as you send in samples and as you run samples within your own facilities. Again, when we talked about the spread plate, very easy to see the colony morphology because everything's on top, and pull those samples. Gives us a better exposure to oxygens for our aerobic organisms. If you're worried about staph, there's staph Baird Parker, and that agar is actually a very opaque color. So if we were to do that as a pour plate, you may not see the colony inside of it, and that's why that staph Baird Parker will always be a spread plate. And again, we talked about what the surface needs to be to adequately spread our organisms across that without letting them swarm. We want to do a larger liquid sample or a sample without particulates. So when we have products such as cheese or spices, this goes out the window. But our liquid samples, we can use membrane filtration and actually pull a large volume of the sample through that membrane and then place it on our desired media to get to our target organisms, our target indicators. So like I said, really limits down on the type of sample that we can run through it, but it does allow us to run a very large sample. So we'll see this a lot of times from a water standpoint. The last one here is the most probable number. So this is where we have a very low population of our organisms, or we have high number of particulates that would interfere on the count on a spread or pour plate, or Petrifilm for that matter. We're going to utilize our serial dilutions here and put multiple tubes from each dilution in place, and then inoculate that looking for some sort of indication, such as that turbidity, where we have that little bit of a color change. Talked about coliforms creating that gassing effect. So you can put a small, what's called a Durham tube inside of the test tube and allow that test tube or the Durham tube to actually rise with that gas production. At the end of it, we get this funny equation of how many tubes changed at each dilution. In this point in time of my life, I've learned that there's calculators and I've been dependent on that. So my math skills allow my hair on the back of my neck to cringe and stand up a little bit when I look at this formula. So thankfully, they gave us this beautiful chart that based on how many tubes we saw that change in and call positive, it'll give us that most probable number of how many colonies or how many bacteria are actually there. Because of that inconsistency though, it does give us that range high and low as well. Final step, or one that that I'm going to just mention here, is our Petrifilm. We do see this largely in the facilities. We do use it largely here at Cherney as well, because of the diversity of applications that it has. I'm not going to go into depth about it or the use, because we do have that November 18th webinar where we'll have further discussions, really looking at the interpretation and use of Petrifilm. So with that being said, let's jump over to the pathogens. Again, common pathogens, we're not going to go in depth on the pathogens, but want to make sure that we're on the same nomenclature, understand a little bit about them and the platforms that we use to get to those, because they do, as we said, have a very prevalent place in our food safety systems, testing our product and our environment. So different pathogens have different ways of being referred to. We can go just to genus, such as Listeria. We can get specific with the species, so Listeria monocytogenes. I'll tell you, the first couple of times that I tried to pronounce that word, no one knew what I was talking about because I couldn't say monocytogenes. So it gets shortened up a lot to Staph aureus or L mono, EB instead of Enterobacteriaceae. And then sometimes we'll start cutting subspecies out. So Salmonella is a big one. There's 2,500 different serovars, only 2 subspecies are subspecies. So Salmonella enterica, subspecies enterica, we get all the way down to where we just look at the serovar and cut everything else out. So instead of saying Salmonella enterica or serovar typhimurium, we'll just call it Salmonella typhimurium. Again, this qualitative, we talked about that seek and destroy. Is it there? Is it not there? So we're going to take your sample, give it all the nutrients, the right temperature and growth environment to really try and get to a detectable limit with even the lowest number of bacteria that were in the original sample. By enriching them and incubating them. Now, again, we're going to look at a video here in a second where it says results within a day. That's once it comes out of the incubator. So just want to preface that by saying we still have the incubation period. And then our results are going to be negative or positive, presumptive if we don't, or before that confirmation step. And it's given in the original sample size. So as our example says here, negative per 25 grams. So here at Cherney, we utilize 2 rapid platforms for that presence absence. And again, rapid as it comes out. We've done the enrichment. Now we want to run and see, is it there, is it not? And there's 2 ways of doing that. On the left-hand side, you can see, or that picture, it's our Vitis unit, and that's looking at the phenotypic method or the antigen antibodies. The one on the right-hand side is looking at our DNA. That's our Bio-Rad IQ check and thermocycler. That's actually looking at the DNA PCR platform, which is that polymerase chain reaction. So to go in a little more depth, I'll quickly explain it. We'll watch a brief video on each of these. Our Vitis is really a lock and key type methodology. The antibody and antigen are going to conjugate, and then a second antibody is going to come in with an enzyme that would fluoresce. So if that whole chain connects it together and makes that sandwich, as that middle picture shows, it's going to start fluorescing light. That light is actually what's being measured. And once it breaks a certain threshold, we can say that there was enough of that organism present, that target organism, to call it a presumptive positive. So have a quick video here to further demonstrate that as well. So if we don't use the antigen antibody type platform for this, we also we'll look at our PCR process. This is what I was talking about with our Bio-Rad IQ check. So this is really looking at finding a way to go in and get a target DNA for our target organism and replicating it to the point where we can see it on a curve to break a threshold. So I'm way oversimplifying this, but basically we're unzipping the DNA helix, looking for that target with a primer to know if it's Listeria, coli, salmonella, and then allowing that primer to attach and start replicating to create that curve. We're lucky to partner with Bio-Rad, and this is actually their video in regards to their Bio-Rad.
Juggling kits and protocols from different vendors? Bio-Rad's IQ check kits provide one simple protocol for all your pathogen testing needs. Short enrichment times and a patented highly sensitive detection method take you from sample to results in one day. How does it all work? Bio-Rad scientists develop patented probes that bind and detect pathogen DNA with unmatched specificity and sensitivity. The simple, real-time PCR-based tests alert you to contamination even before the reaction is done, while AOAC and AFNOR validation ensure peace of mind. Testing hundreds of samples, the IQ Check prep system automates sample prep and is the perfect solution for higher throughput labs. Regardless of your testing volume, Bio-Rad has a simple and efficient pathogen detection solution.
So here at Cherney, we do have that whole IQ robot to allow for a little more hands-off. Obviously, we always talk human errors and ways of taking that off. We have great technicians here, but this allows them to run more samples in less of a time period, get you the results quicker, and like I said, a little more hands-off. So great opportunity. Two very great platforms using different technologies to be able to give you a lot of information quickly about your environment, your product, from a food safety and quality standpoint. So switching gears one last time here, I just want to mention biofilms and how that plays into our pathogens and our indicators. So many of you that have dealt with biofilms may have cringed a little bit when I said that word, because that is that bacterial attachment to the surface using extra polysaccharide to really protect it, create a nice little home for it, and allow it to be able to survive basically our sanitation when we talk about that washing, cleaning, treatments, biocides. So our very quick depiction of it is we can see here on step one, we have bacteria floating around, getting to the point to 2 where it's going to start attaching to that surface. And by the time we get to 3, it's already in protection mode, where that it's not going to necessarily come off with regular sanitation. May need that little extra elbow grease in there. It grows into the point where the bacteria don't have enough room, and then it expels them back out. And now also we're starting to see, hey, we have this target organism back in our environment. I mention this because sometimes our indicator organisms will actually play nice with our pathogens and give them protection as well. So one of the common ones that we talk about in water as an indicator organism is our Pseudomonas. Pseudomonas is a bacteria that will create a biofilm and play nicely with our dear friend Listeria, granting Listeria that protection from their biofilm. So as you look at what you want to test for indicators in your water, in your environment, just keep in mind that these biofilms will also play another important role to be able to kind of tell you what's going on in your environment. So I know we covered a lot here in a little bit of time at a pretty high level. If you have any more questions or you need this testing, please don't hesitate to give me or any of our team members here a call or an email. We'd love to continue on the conversation. We're going to stick around and do a Q&A session here after, as well. But if you got to get going or you have these questions that arise later, please let us know and continue the conversation. I'd mentioned we do have 3 additional webinar educational opportunities coming up. So the next one is our good lab practices and safety, which will be October 7th. Then we're going to go into aseptic technique in late October and finish out in November, as I had mentioned in the presentation, with Petrifilm use and interpretation. So with that, thank you guys very much for participating and being a part of this today. So happy that you can make it. And I'll open the floor up with our team here for any questions that you have. And I do see we have a couple things coming in here. So I see the question here is, where in your opinion is the line between an indicator and a pathogen? What supports your opinion? So Mike, I guess just to clarify on that is when we're looking at the Enterobacteriaceae family and you have E. coli in the coliforms or you have Salmonella in the EB family, is that, if that's what you're referring to, theoretically, absolutely one colony on an EB plate could be Salmonella. We know that there is that risk, but many of these other organisms will start growing before that and give us that indication of that growth environment. So that's kind of where we cut that line, knowing the risk, but still using that indicator within our environment. So Allison, I think, was looking at Pikachu. All right. Allison was looking at the dilution factors and the diluents to use. Oh, so Listeria species as an indicator test, like I had mentioned, similar to the EB, that Listeria monocytogenes is the wimpy kid. So in the environment, definitely be looking at using Listeria species just to know that the environment's correct. Now, when we get to our product, that's where we may want to look a little more in depth at seeing that as a Listeria monocytogenes test directly. The opportunity is always out there to go into speciation when we do a Listeria genus test. So if you start seeing something that may indicate a Listeria problem or Listeria growth, and you're worried about it being monocytogenes, I would strongly suggest taking that to the next level and going through confirmation and speciation. So if you're testing for EB, you are actually testing for the coliforms. That's all going to be included in that family. So you will see your coliforms. It's taking it to that next level. So no, you would not need to do necessarily an EB and a coliform test. The bacillus. Okay. I see another one for bacillus, and then there was a question that I thought, what would cause a product to come back presumptive and then come back as negative. So there's a couple of things based on the platform that you're using. The PCR platform will pick up dead DNA, or what we call non-viable, non-cultural DNA. So if you have that present, it's not going to confirm out because it's not cultural. The DNA is technically, I'll call it dead, but still existing in the environment. So it's just finding it still there in the environment. There's a couple of opportunities to that. There's free DNA removal solution that can be added to your sample and then rerun on to kind of eliminate out that DNA. On the Vitis, because it's a lock and key, with Salmonella especially, there's a couple of different organisms that have the same key to the lock. So that is, if we see it coming back as negative on our confirmation or not confirming, that may be what we're talking about, is one of those other bacteria that have that similar antigen connecting on and showing us that it broke that curve or broke into that curve as a presumptive. Okay. And Allison is covering the difference between CFU and MPN. Any other questions that we didn't answer today? All right, well, we'll stick around for just a couple more minutes in case there is some other questions. But again, I strongly encourage you, if something comes up or you have questions that you don't want to ask on here, to give us a call. Allison and Michelle are back here. We have a couple other members of the technical team that are phenomenal resources, as well as our client services team being able to answer many of those questions as well. So thank you guys all for your time today, and I hope this was of benefit to you, and we hope to see you October 7th, or well, virtually, I guess, see you on October 7th.